All right, welcome back to the deep dive. Today we're diving into ADCs, antibody-drug conjugates. As always, we're going a little bit deeper.
That's right. Deeper into the methods, I think, would be a good way to put it.
Yeah, kind of the experimental nuts and bolts.
Yes.
And really trying to understand the logic of how these experiments are designed and what they can tell us.
Exactly, because as you know, the idea of using an antibody to deliver a really potent toxin specifically to a cancer cell, that's been around for a while.
Right.
And there are a number of approved drugs now.
Mm-hmm.
But the challenge really is, how do you select the best antibody, the best toxin, and then link them together in a way that is stable?
Right. And then you have all the in vivo challenges, like how does it behave in the body and does it actually get to where it needs to go?
Exactly. So this paper we're going to be talking about today, they've developed a really nice platform. It's elegant in its simplicity, and it's quick and cost-effective.
And we'll get to exactly how it works. But first, I think it's good to lay the groundwork. Why do we even need this kind of rapid screening approach? Like, what are some of the limitations of more traditional methods for developing ADCs?
Well, you bring up a really good point. ADCs have shown tremendous promise in the lab and in the clinic, but unfortunately, there's a pretty high attrition rate. A lot of ADCs that look good in early studies, they just don't pan out in later stages.
So it's like we need better ways to sift through all the possibilities early on, right?
I mean, if you think about it, you have all these different antibodies, you have all these different toxins, you have all these different ways to link them.
Mm-hmm. It's a combinatorial nightmare.
It is. It's a huge combinatorial problem. And the traditional methods for linking antibodies and toxins, they can be really laborious and time-consuming.
So by the time you get to the point where you're testing something in vivo, you've already invested so much time and resources.
Exactly. So this platform that we're talking about today, it takes advantage of the really strong interaction between streptavidin and biotin.
Right. Streptavidin and biotin, for those who aren't familiar, it's a very well-established system in molecular biology. Like super high affinity, they bind really tightly to each other.
It's almost like a lock and key. They're not covalently linked. It's a non-covalent interaction, but it's so strong that it's essentially irreversible under physiological conditions.
Right. And it's interesting because streptavidin and biotin, it's already widely used in all sorts of applications like labeling, detection, purification.
You're exactly right. It's a real workhorse in the lab. But what's new here is that they're using it to very rapidly generate these antibody toxin conjugates for initial screening.
Okay, so walk us through the platform. Like, how does it actually work in practice?
Sure. So the first part of the platform is focused on selecting antibodies that are internalized into the target cell. And this is key because for most ADCs to be effective, the antibody-drug conjugate has to actually get inside the cancer cell.
Makes sense. You need the toxin to be delivered into the cell to do its job.
Right. And to do this, they use saporin. It's a toxin that inactivates ribosomes, which are essential for protein synthesis. So saporin is super potent, but it can't cross cell membranes on its own.
So it's like a Trojan horse, the antibody is the horse, and saporin is the soldiers hidden inside.
Exactly. So if you see cell death when you treat cells with an antibody-saporin conjugate, you know that the antibody was able to deliver the saporin inside.
It's a clever way to indirectly measure antibody internalization.
Right. And the actual process is really straightforward. They first label the antibody with streptavidin,
which takes about 3.5 hours, according to the paper.
Yeah, that's right. And then they conjugate the streptavidin label antibody to biotinylated saporin.
And that's a much quicker step, only 15 minutes.
Yeah, it's super fast. And they found that on average, about 2 streptavidin molecules get conjugated to each antibody.
Okay, and what kind of results did they get with this system? They started with trastuzumab, right? Targeting HER2.
Yep. So they showed that even with the streptavidin and the biotin saporin attached, trastuzumab could still bind to HER2 on SKBR3 cells, which are HER2-positive breast cancer cells.
But they did need to use a slightly higher concentration of the conjugate to get the same level of binding as unconjugated trastuzumab.
Yeah, which you might expect. I mean, you're adding all this extra bulk to the antibody. It's going to affect the binding kinetics a bit.
Right. Steric hindrance, as they say.
Exactly. But the key finding was that the trastuzumab streptavidin biotin saporin conjugate, or trastuzumab deruxtecan, as they call it, was really effective at killing HER2-positive breast cancer cells.
Both SKBR3 cells and HCC 1954 cells, which are another HER2-positive cell line.
Right. And importantly, it didn't kill MDA MB231 cells, which are HER2-negative.
So it's very specific.
Very specific. And to really show that this wasn't just a fluke, they tested a bunch of other antibodies and cell lines.
So an anti-CSPG4 antibody in melanoma cells, cetuximab in EGFR-positive breast cancer cells, and MOVT in FLAIR-positive cells.
Right. And in all cases, they saw the same pattern, target-specific cell killing. The conjugates killed the cells that expressed the target, but they didn't kill the cells that didn't.
And just to be clear, neither saporin alone nor the naked antibodies without saporin had any effect on the cells.
Right. So it really demonstrates that the saporin is being delivered specifically by the antibody.
So the first part of the platform is all about identifying antibodies that are internalized. And the second part is about testing different payloads, right?
Exactly. Once you know that you have an antibody that gets inside the cell, you want to see what kind of toxin will be most effective.
And to do this, they basically biotylate the toxins so that they can be conjugated to the streptavidin-labeled antibodies.
Right. And they use toxins that have either an anamine group or a thiol group, which are common targets for linker chemistry in traditional ADC development.
So they're using the same kinds of chemistries that are used to make clinical-grade ADCs.
Exactly. And to really show that this approach could work, they compared their system to trastuzumab emtansine, or TDM1, which is an FDA-approved ADC.
So TDM1 consists of trastuzumab linked to DM1, which is a microtubule disrupting agent.
Right. So they took DM1 and biotinylated it using its thiol group.
And then they conjugated the biotinylated DM1 to streptavidin-labeled trastuzumab, and they aimed for a drug-to-antibody ratio, or DR, of about 3.5.
Which is similar to the DAR of TDM1. And just to explain, the DR is basically the average number of drug molecules that are attached to each antibody.
And it's an important parameter because it affects both the efficacy and the toxicity of the ADC.
Exactly. So they made their own trastuzumab SBDM1 conjugate, and they compared it to TDM1.
Head-to-head.
Head-to-head. And what they found was that their conjugate had very similar binding kinetics to both trastuzumab and TDM1.
When they tested it on HER2-positive HCC 1954 cells.
Right. And when they looked at cell killing, both TDM1 and trastuzumab SBDM1 were equally effective at killing HER2-positive cells.
Again, no effect on HER2-negative cells.
Right. So they were able to recapitulate the activity of a clinically validated ADC using their rapid conjugation system.
Which is pretty impressive.
It is. And to show that this approach wasn't limited to just trastuzumab and DM1, they also tested a different antibody, MOV18, which targets FRI.
And they used a different payload, A419259, which is a SARC family kinase inhibitor.
Right. So they biotinylated A419259 using its primary amine group, and they conjugated it to streptavidin-labeled MO18.
And they found that the MO18 SBA 419259 conjugate was very effective at killing FRA-positive cells, but not FRA-negative cells.
Again, showing the target specificity.
So it seems like this platform is pretty versatile. You can use it to test a variety of antibodies and payloads.
That's right, as long as the toxin has a functional group that can be biotinylated.
Okay, so they've shown that they can make these conjugates quickly and that they can kill cells in a target-specific manner. But do we know how they're actually working? Like, do they have the same mechanism of action as conventionally produced ADCs?
That's a really important question. And to address it, they used confocal microscopy to visualize what was happening inside the cells.
So they treated HER2-positive HCC 1954 cells with either trastuzumab SBDM1 or TDM1.
And what they saw was that both conjugates were internalized into the cells.
So the streptavidin biosyn linkage didn't prevent the ADC from getting inside.
Right. And they also saw that both conjugates caused the microtubules to depolymerize.
Which is how DM1 works. It disrupts the microtubule network, which is essential for cell division.
Right. So this suggests that the trastuzumab SBDM1 conjugate is working in the same way as TDM1.
Delivering the DM1 payload inside the cell, where it can then exert its cytotoxic effect.
Exactly. And they didn't just stop there. They also tested their trastuzumab SBDM1 conjugate in vivo.
In mice with HER2-positive human breast cancer xenografts.
Right. And they found that a single dose of the conjugate at 4 milligrams per kilogram was really effective at inhibiting tumor growth.
Compared to the control groups, which received either a buffer solution or naked trastuzumab.
And importantly, the tumor growth inhibition was comparable to what they saw with TDM1.
So the rapidly conjugated ADC was just as effective as the clinically approved ADC.
That's right. And they didn't see any signs of toxicity in the mice. They monitored body weight throughout the study, and there were no significant differences between the groups.
So it seems like this platform has a lot of potential for accelerating ADC development.
It does. I mean, it's fast, it's cost-effective, and it seems to generate conjugates that behave very similarly to conjugates made using traditional methods.
But the authors are very clear that this approach is not meant for clinical use.
Right. And there are a few reasons for that.
Like, streptavidin is a foreign protein, so it could trigger an immune response in patients.
Yeah, that's a big one. And the streptavidin-biotin linkage also adds a lot of bulk to the ADC, which could affect its pharmacokinetics.
And the linkage is non-covalent, so there's a chance that the conjugate could fall apart in the body.
And there's also the issue of endogenous biotin. There's biotin circulating in our blood, and that could compete with the biotin-related payload for binding to streptavidin.
Right, so it's really a research tool. It's not meant to be used in patients.
Exactly. But it's a really valuable research tool. It allows you to screen a lot of different antibody-toxin combinations very quickly and efficiently.
And identify the most promising candidates for further development using more traditional conjugation methods.
Right. So it's a way to streamline the early stages of ADC development.
So, in a nutshell, this platform uses streptavidin biotin conjugation to rapidly generate ADCs for preclinical screening. It allows you to test both antibody internalization and payload activity in a relatively high-throughput manner. And while it's not meant for clinical use, it has the potential to accelerate the development of more effective ADCs.
That's a great summary.
Now, for our listeners, I want to leave you with a thought. We've seen how powerful the streptavidin-biotin interaction can be for this kind of application. But are there other biological interactions out there, maybe even stronger or more specific, that could be leveraged in a similar way? And thinking about the limitations of this specific platform, what kind of creative solutions could be developed to overcome those challenges and make these screening platforms even more robust and predictive?
Those are fantastic questions. And I think the answers could have a major impact on the future of drug development.
Absolutely. Thanks for joining us for this deep dive. We'll see you next time.